We've got an amazing jam-packed program already outlined for the year for our monthly meetings, which hopefully you'll enjoy a lot, including this afternoon's talk by Arash Zamyadi. I'm going to start with an acknowledgement of country. I'd like to acknowledge the traditional owners on the land on which we meet, wherever you may be joining us from today. And we'd like to pay our respects to the elders past, present, and emerging. I am going to just acknowledge the support we get to run EnviSMART. We really appreciate it. We're supported by Agilent, Eurofins. We're partnered with different organisations that include the Australian Contaminated Land Consultants Association, Water Research Australia, and CTAC, the local branch, and also the Asia Pacific region. And we also have some other supporters too of Trajan and PM Separation. So really appreciate all the support we get from the different groups. It really helps us to be able to run these events each month. And I think, as I mentioned, we've already got this amazing program lined up for you for the rest of the year. So notionally the meeting's on the third Thursday of the month. One's been moved, the one just before Easter, because it's going to be the second Thursday of the month. But we've got a talk on mercury next month by the current president of Asia Pacific CTAC. Following that, we have a talk on statistics, which for every environmental person, you probably should come and hear this talk. He's Dennis Helsel. He's written a lot of articles on sensor data in particular, and he's from the USGS. We're going to have from Europe, Carolyn Mormont, who has written quite extensively on best practice for running ecotox experiments. In June, we're going to have Michelle Hadlick from the USGS as well, talking about neonicotinoid insecticides. And in July, we've got Jenny Stilber from CSIRO joining us. And I see Kath just jumped in. Hey Kath, how are you? I don't know if your audio looks like it's still connecting. So that's what's coming up this month. Well, without further ado though, I'm going to introduce our talk tonight by Dr. Arash Zamyadi. Arash, I don't have your bio, I'm sorry. But Arash is going to tell us all about algal blooms, and he's currently works for Water Research Australia, and he's also a senior lecturer at the University of Melbourne. Thank you very much, Arash.
Perfect. Do you see my screen, Brad?
Got it. And we can see you as well.
And do you hear me properly?
Yeah, all good.
Perfect. Thank you. First, I'd like to thank Brad and EnviSMART team for giving me this opportunity. Thank you so much. I much appreciate the invitation. And hello, everyone. Thank you for joining us tonight on this presentation. So as Brad mentioned, I'm Arash Zamyadi. I'm Water Research Manager based in Melbourne, hosted by Melbourne Water, and also hold a senior lecturer position in chemical engineering at the University of Melbourne. So what we're going to talk today is about algal management. What is the problem? How can we detect them? And how can we treat them from the water? So let's have some algal fun, which looks like this picture, a good substantial bloom. So just a bit of background. These cyanobacteria, as you can see the picture up left top, is an algal bloom. You can take this bloom and put it under a microscope, and you can see the cells. This is the picture. You have two species. You have Microcystis and Anabana, what used to be called Anabana. It's now called Dulcaspermum. I can't pronounce the name properly, but it used to be called Anabana. When you put them under a fluorescence microscope, that's why you see them, they're happy, healthy cells. And under electronic microscope, you can see the individual cells. If you zoom in more, you can actually see a full cell by itself. They look like minions, don't they? Minions kill dinosaurs. Cyanobacteria probably killed some dinosaurs as well, by the way. They were found beside the fossils of dinosaurs in Brazil, their fossils. Well, what is the interest to us is what is inside. So these red dots are metabolized organic matter that are inside the cell. and the metabolite or organoid that is outside the cell. Keep that in mind. I'd like to show this picture at the beginning of the talk to visualize what is inside and what is outside. Very important for treatment, particularly, and detection. So, why are we worried about these algal blooms, the cyanobacteria blooms? First of all, they can cover our water bodies. They can cause lots of problems from light to nutrients, to access to oxygen. As you can see, they can go up and down in water bodies. So about 70% of the body of a cell of a cyanobacteria is full of gas vesicles. So it allows them to go up and down. So in the day they go get the sun and they go down and then the nutrients can come up. That's why they can survive in different environments and also overtake lots of other phytoplankton who live in the water. Why are we worried about them? They produce toxins. So everybody knows probably what microcystin is. If you want to have an example, microcystin is like Brad Pitt. When I say Brad Pitt, everybody knows Brad Pitt. But there are other toxins who are less known, who are like the new kid on the Hollywood Boulevard, who wants to become Brad Pitt. So they can do nasty things to become famous. One of them is like anatoxins. For a long time, we thought we don't have anatoxin in Australia, but now we know we have. And then there are other toxins that there are lots of unknowns involved about them, like BMAA. The point is that there are thresholds, WHO, health organizations across the planet, including Australia. What is getting interesting is that in North America, now they're talking about thresholds for bottle-fed infant. You know, in a household, there is not, I have one at home, he's not bottle-fed anymore, he's 4 years old, but there's no separate tabs, one for daddy and one for the kid. So we have to adapt to the one that is for the kids. So that's about the toxins. Then it's about taste and odor compounds. Taste and odor compounds are not toxic, but studies have shown that people associate them to water being unsafe. So, and they're unpleasant. So no customer likes that the water tastes earthy, musty, and they're very hard to remove from the water, and humans can detect them in very, very low concentrations. The other challenge is this: you can find them anywhere else on the planet you want to. Look at those black dots on the planet. Those are the places that we have detected cyanobacteria. And also look at that, Maury darling, catchment, and look at the dots and the blooms. Everybody knows about the 2016 famous bloom that covered over 1,600 kilometers of the river. So when you look at our water supply system, that I put a picture down here of it, they can be found in our water resources. We will talk to you and you will see the breakthrough into our treatment processes and into a distribution system. And they go all the way into our wastewater treatment system, recycle system, and go back into the environment. And by the way, if I was about this guy, those fish died from probably lack of oxygen. But this dog, which will probably take a bite of the benthic associated with this bloom, I would be more worried about this dog. One thing that everybody focuses on again was that everybody focuses on the Brad Pitt, so microcystis, which produces microcystin. Everybody focuses on the most famous of them. When you go by the present, we did a study that we published recently. We looked at the blooms across the globe. When I say across the globe, I mean where there was a published record, a reliable publication. And what you can see is that when you look at the number of times we've detected presence of cyanobacteria, obviously microcystis, which produces microcystin, comes as the one that is most important one. But when you look at the cell number, so actual cells that are present in the water, you can actually see there are other species like Aphanesomnon, like Clandosperm mopsis, which they produce Clandosperm mopsin, that actually they can be as present in numbers as much as microcystis. It's important to keep in mind there are different species of cyanobacteria with different characteristics, including different filamentous form that's present in our water. And when you look at this, these are pictures from a treatment plant in Australia. And you can see they can break through a treatment system. This top picture is a mixer. Here, normally you mix your coagulant. You don't want such a green carpet on top. And then they accumulate in our sludge, which is a good thing. You can see them being accumulated in the sludge. But what is more surprising to us is in recent work that we have done, and we published it about both anatoxin and BMAA. About anatoxin, for a long time, we thought we don't have anatoxin in Australia. It's a neurotoxin, but now we've found benthic producers of anatoxin. And BMAA, there's lots of questions involved about this neurotoxin and its association to algal toxins. So one must take it with a grain of salt, but there are lots of concerns involved. So First, we need to detect these cyanobacteria. So what I'm going to talk about, I'm talking about the monitoring pathways and also early warning systems. Let's jump into monitoring. To monitor these cyanobacteria, one way is to go take a sample, put the sample on the microscope. It takes a while. You need to have a trained eye. Why not use fluorescence probes? Cyanobacteria have their own a specific signature pigment called phycocyanin. So we can detect the fluorescence of phycocyanin, which will tell us if we have cyanobacteria or not. It will not tell us which species we have, but it will help us to say that we have cyanobacteria. And it also will help us to follow the trends, if we have too many of them or less of them. Then there are methods. We can take the same sample. There are new methods like FlowCam, which helps us. It's a combination of a microscope and a flow cytometer, and it helps us, and a particle sizer, by the way. It helps us to identify which species we have. What is getting more interesting is imaging techniques, very similar to artificial intelligence techniques to use to identify human face. Those techniques can be used to identify cyanobacteria species. So that is emerging, and it's really interesting. Once you confirm that you have these cells, that is when you need to see if I have metabolites. So either you can take a sample and run the sample through GCMS, for example, which I'm not going to talk about that because we have the absolute experts of that, Brad, here. So please talk to Brad if you have any questions about that. He's the kind of the god on this matter. And then, or you can take the sample and run PCR, look into the gene responsible for the production of the toxins or taste and other compounds. The presence of the gene doesn't necessarily mean that they will produce this, but it at least tell you if they have the potential or not. Then you can use fast kits like ELISA kits to see if you have production of toxins or not. And then again, use methods like LCMS, which Brad is still the expert again here. We can run to measure the concentration of the toxins in the water. This is a very, how do you say, summarizing the way to detect these cyanobacteria. But utilities are interested in early warning system. What we decided to do with water in a recent project with Water Research Foundation, sorry, I don't know why this slide is coming blurry, but apologize. It doesn't look like on my smaller screen, but here it is. And is that we have classified these into 3 classes. First is to just identify the activity of the bloom. You can use things like ATP because it will tell you if there is a change in the overall about microbiology activity in the water. You can go after chlorophyll A, or because it tells you about the entire phytoplankton community, optical density, visual inspection. You can use things like the color of the water, the CH depth, or the turbidity. Then tier 2 are the ones that are specific to cyanobacteria, like the phycocyanin. You can take a sample on the microscope, or you can then use it for gene detection, which will only tell you about the cells and the capacity to produce toxin. Tier 3 is that once you know you have the cells, then you can run for toxin, taste and other compounds, and also use again a specific method targeting a specific toxins or taste and other compounds. One thing we said before we go further, let's talk to utilities. So we ran a survey of 34 utilities in Australia, Canada, and United States. And what we sat with them and we talked with them, survey was open to all the utilities members of Water RA and Water Research Foundation. We got hold of 34, which we really appreciate their input. And what we did, what we're trying to understand was, interestingly, 10 of these utilities didn't even have any plan to deal with cyanobacteria. None of them were in Australia, by the way, so that's good news. But we were interested to know what methods they use. As you can see, most of them use the traditional one. They look at chlorophyll, they do for cell counting, and then they go for taste and other compounds or toxins. One other interesting thing we did was that we classified their perception, their point of view of these methods. So these color codes here, red means that they were not happy, yellow means it's a so-so, and green means, oh, okay. What is not about again exact science, is about the result of the survey. And you can see here, it's very interesting how, for example, they found visual inspection, which is true, has lots of interferences, and the accuracy is not high. As you can see, they still found one of the reference method, which is the cell count, the one that has the lowest interference and the highest accuracy, but it takes fairly long time to get the result. One thing I want to pay attention here is the satellite. It's emerging using satellites, but you can see from utility point of view, they think accuracy is low and the loss of interference involved, particularly in the cloud cover. So that gives us a good understanding of where utilities sit and what their needs are. So that brought to Water a recent project that we built that in collaboration with Water Research Foundation. We said, okay, imagine we use these methods to detect the gene, the DNA responsible for production of toxins, which actually there are methods, the standard methods available for it. The question utilities asked is that how can I integrate that into my management plan? These use of gene testing methods are regulated in places like the states of Ohio, Oregon, and Los Angeles has regulated that. A city in Texas has put that into their management plan. State of Massachusetts is using that. Indiana is using that. But what about in Australia? What are we doing about that? Our utilities, lots of them actually take samples for detecting the gene responsible for production of the toxins, but it's not integrated into management plan. So this project is trying to help how we can help these utilities integrate gene testing. For example, a good example of that is what Ohio is doing. Ohio is actually using the gene testing before even they take samples for cell counts. So remember, based on the survey, most utilities trust the cell counts, but cell counts are hard to do. You cannot take so many cell count samples, but with lots of real-time qPCR, you can take lots of samples and you get the results fairly fast. The state of Ohio has decided to actually incorporate use of gene testing methods into the management methods. One thing we need to really take into consideration is the presence of the benthics. We have traditionally focused only on the plastic ones, the ones that are within the water body. We don't pay much attention to the one that are on the sediment. And as I mentioned, when we found anatoxin for the first time in Australia, it was found that it's hypothesized that it's mainly produced by the benthic cyanobacteria. They're hard to sample, they're hard to monitor, but we're seeing more and more their presence and their impact on production of taste and other compounds and toxins. So another project we have, you remember on that pathway, I said we can measure the signature pigment of cyanobacteria, which is phycocyanin. You can measure that on real time. So we have a project with Water New South Wales and Valley Water from California, which they have installed the probes at their intake, so when the water comes into the plant and throughout their plants. I'm giving here an example of the system provides value to Silicon Valley. And we are looking into how these fluorescence measurement probes can integrate into their management. What do they mean by integration? Here I'm giving you an example. Here you have the probe readings over the year 2019. You can see the blue line shows the fluctuations only from the probe readings. So this is ratio fluorescence unit. So it measures the pigment. It doesn't measure the cells, but the up and downs tells you if you have cyanobacteria or not, or how much of phycocyanin you have. And then you can come up with the red line as the management. So we correlate that red line to the management threshold, which was on cells per ml. And we came out with the threshold which says if your probe measurements are above that threshold, you should do something. You should follow your action plan. But are these peaks accurate? The loss of source of interference involved with the measurements of real-time measurements of fluorescence. So what we did actually, we did a study in Lake Erie because we had to access a bunch of probes in Lake Erie, actually 75,000 data points. And we used that to train an algorithm to see the anomalies involved with cyanobacteria phycocyanin reading. And as you can see, based on this study, the red dots are the peaks that were not correct. So you can see that there are still lots of problems involved with using real-time measurements of phycocyanin, but we have tools that they can help us to understand which peaks are correct and which peaks are not correct. Despite all these problems, utilities are seeing benefits in using these probes, including SA Water. SA Water use these probes not as to absolute measure of concentration of the pigment or the cell per ml, but as a trend. So they see they have established a baseline. When there is low season, they have no cyanobacteria problem. So they know where the baseline is. And they use the probe to see when the trends to start go up. And when the trends to start go up, they trigger intervention. They start to sample more frequently. They start to look into what type of hydroactivated carbon or coagulant they have to do, or what type of source mitigation they have to implement. The other method that people are looking more and more is use of satellite for remote sensing. As I mentioned, utilities have attempted to use them, and they have questions and concerns about them. Hence, Water RA had the project in collaboration with Melbourne Water, Icon Water, and Water New South Wales, working with CSIRO Aqua Watch team into looking into, okay, let's first look at what are the advantages and what are the limitations of remote sensing using satellites, and what information we can add to them. We're going beyond algae or fluorescence because we're even using them for fire spotting. But one of the benefits of this using remote sensing is mainly to measure the fluorescence. So now let's say these methods that are listed, from real-time detection of fluorescence to detection of genes, detection of the toxins, and taste-and-odour compound work. And they tell us we have a problem somewhere within our supply chain, from source to the treatment. So what do we do with them? As drinking water or water utilities, we try to treat them. I'm going to talk about three options. One is about source management. One is about try to tackle the metabolites, the toxins. And one is about to get rid of the cell. Do you remember I started this conversation by saying I showed you the minion, the cells, and I put that red dots, the one that are inside the cells and the one outside the cells. Options A and C are looking at take care of what is inside the cell with the cell, and option B is looking at what is outside the cell. And I will explain why I listed them at these A, B, C options, but you can sort them around. So option A is source management. One of my detection methods that I have told me that I have some major cyanobacteria problem. I have to do something about it. There are different ways we can do. We can rely on other lakes or reservoirs if we can change our intake. That is really dependent on if we can change the intake, if we can change the depth. If we have fluorescence probe, we can see where the bloom is located and change the depth of the intake. We can go toward river supplies. River supplies, because of the current, normally they have lower cell numbers. We can look into solutions like riverbank filtration, even though we have published papers that show that there are breakthrough of cell to riverbank filtration. Or we can use groundwater supplies that are far enough, because the recent studies, particularly from Lake Tahoe in China, showed that penetration of live cells into groundwater systems about 50 kilometers away from Lake Tahoe. So these are solutions, but still you have to have consideration. What I like to show here is the schematics, and I think Simon Tannick will appreciate this schematic. That one thing you can do at the source is about taking care of the nutrients. So we add lots of nutrients, phosphorus and nitrogens, that in different forms that are biologically available. If we can find ways to lock these, so they are not accessible to our cyanobacteria or we change the structure of water body in a way that other microorganisms in the water consume them and they won't get to the hands of cyanobacteria. That's one way of controlling source control. The other ways that our people have looked into them is things like sonication, which I will talk a bit about more, about mixing. The mixing will help to enhance the grazers and other microorganisms to live in the water. Biological control is another way to look at it, and biological manipulation. And then there are other ways to try to make these nutrients non-biologically available, and maybe also remove the sediment. I was told that if we even stop tomorrow, the loss of our reservoir, the inflow of nutrients, we will have enough nutrients accumulated in the sediments for many, many years to come. So maybe removing these from our water bodies is one way of looking at it. But when I come to sanitation, we have developed actually a project that is looking into developing protocols for utilities to test these different technologies. And one of the technologies that was flagged to us by Intelligent Water Network and some Victorian utilities was sanication. So how does sanication work? As I mentioned, about 70 or 80% of a structure of a cell of cyanobacteria is gas vesicles. And that's what allows them to go up and down. Imagine you find a way without breaking the cell membrane, we burst those gas vesicles. This way the cell will drop to the sediment and they will die off. So this way we will get rid of the cyanobacteria. That's what sonication does. The lots of questions involved about sonication, particularly it has been used previously in very large waters of body. So what we have done with support from Victoria EPA, Central Highland Water, Intelligent Water Network in Volia, we are testing 2 sonicators in a smaller reservoir in Central Highland in Victoria. Also, apologies, I don't know what happened to my window. Also, we have installed now 4 of them due to preliminary result that we saw in Central Highland. 4 of them are now being installed in Crongamite Shire in a reservoir that is used for recreational activity. I'm just going to show you some preliminary results. So these are preliminary results from some initial work we've done. We haven't run proper stats on them yet because we are still waiting for this bloom season outcome. But what we can see here, what you have on the vertical line, and I put the picture of the sonicator, is that it was about December 2020, we installed the sonicator at the Central Highland Reservoir. And you can see here, prior to that, we have the cell numbers. There was still not very high cell number. You can see there were only few occasions that they went over the management threshold. But what I want you to see is that, for example, the fate of microcystis, that how we are observing a drop in number of microcystis that were detected in water, or a drop, for example, in number of Pseudoanabaena. So as I said, we are still waiting to do more stats on them, but they seem to be a promising way to look forward. There are other methods of biomanipulation that can be used in water bodies that we have ongoing projects on them. So hopefully we'll be able to talk more in future as those projects complete. So now, you might say that, oh, I don't want to I have a reservoir or I have an intake that I can oxidize. So then one other way, the option B, is actually to say, why don't we just actually attack the cell, burst whatever is inside the cell, and then take care of the dissolved compound. This way, at least we won't have the cells accumulating in our plant and have that problems, as I showed at the beginning, those green carpets inside our coagulation process. We take care of that, but then we will have dissolved compounds. Saying that, we observe something very interesting. Oh, sorry, those red circles have moved. But what I'm showing here, you see here cells of Cyanobacteria. These are cells, long filaments, and these were all ozonated. What I want you to see here is that where the red circle is, you can see between the 2 cells, there was an empty space. We brought that and looked at it under microscope and saw actually this empty space was the cell that was completely ozonated and destroyed, but the cells that are each side seems intact. So that idea of peroxidize and get rid of the cells and release everything and then take care of the released compounds, this is a bit questionable because ozonation is very strong oxygen agent, and we had a large CTs here, and we are still seeing cells under microscope. This is again another example I'm showing of Microcystis aeruginosa and Anabaena philosacui, that you can see here the pre-oxidation one, A and C, happy healthy cells on the electron microscope. I was expecting post-oxidation with these CTs of both ozone and chlorine, which are high CTs. I wasn't expecting to see any cells. We are still seeing cells that are damaged, or we are seeing these cells. What we observed was that maybe as you do cell oxidation, because their cells are available, when you're oxidizing your cell, you are dealing with whatever is attached to the cell and whatever intracellular compounds you have. So you oxidize the cell, and as you can see, you might damage the membrane, what is between the 2 cells, or the organic matter that is loosely attached to the cell. So your oxidant agent will actually become consumed by these competing material that are released into the water. And this has led to a new project that we are doing under the leadership of Professor Linda Blackall at UNI Melbourne, is that we're looking at epibiomes. So what are other microorganisms who live with the cyanobacteria, adjacent to them, in between close to their membrane, that might actually take up the oxidant agent and consume that oxidant agent, so there will be none left to take care of the released toxin. So that's an ongoing project. Besides on that, we're also looking at the impact of that these epibiomes on the formation of biofilm on our biological activated carbon for removal of released compounds. So the message here, the clear message, is that when you are oxidizing to release the material from the cells, we always have focus on oxidation of released compound. If you remember, this is the red dot that was outside the cell, but there's always this process of taking care of the cell. Here you can see these were compared between the lab cultures and a bloom from in Canada. And you can see when you look at the lab culture, because we had enough oxidant agent, it was pure culture, there was no other competing material. We were able to bring the intracellular and extracellular compound to the same level. But in the real world, which we had organic matter background from the water, we have other microorganisms that were associated to bloom, we actually can see that not all the toxins inside the cells were released. For this difference between the two lines is what has stayed inside the cells, because our oxidant agents weren't consumed by other compounds in the water. So this brings us to this schematic, which is about using source oxidation, because you can use, we do not encourage use of copper sulfide because copper still is heavy metal and toxic for the environment, but one can use hydrogen peroxide. A stabilized hydrogen peroxide is very good. It targets cyanobacteria, and the byproduct of oxidation is water and oxygen. So, but it is very important to keep in mind that when you use this method, you will compete, your oxidant agent will compete with other material, other competing material in the water. And this is a table that summarizes the fate of different toxins. So once the toxins are released, then this table tells you what oxidant agents do to them to remove them. We thought ozone is the magic, ozonation is the magical oxidation that will remove, take up all our toxins. It's fairly correct to say that for most of our toxins, but not for saxitoxins. That was very surprising, that ozonation is not efficient for removal of saxitoxins from water. So if you have different species in the same bloom, which is possible, and if you have microcystin producers, you are safe with ozone. But if you will have saxitoxin producers, then you need to think of something like chlorination. With this outcome was very interesting. So I really, I love this table, because it really tells us how you can have competing targets and how you have to actually mix and match different oxidant agents to remove different toxins. The last option was to remove the intact cells. So one would say, Arash, you know what? I'm not going to do any source mitigation. I will have the bloom in my reservoir. It's anyway seasonal. It will have once in every hot summer, and I will allow it to break into my treatment plant, and I will be able to remove it. I'll do the treatment process on time. I don't want to do a pre-oxidation. So I don't want to release the intracellular compounds. I want to remove these cells that occasionally break into my plant. I'm all for it. Let's do that. But what is important is what happens to these cells once they accumulate inside the sludge. So there has been two other research foundation projects that we've involved with, that it looks at not only what happens inside the sludge, also what happens once you take the sludge out of your treatment supply system, and what you do with that sludge, which will have impact on the use of a sludge after your treatment. What was really interesting, we did some genomic works, and we looked at it's very hard to do cell counts on the sludge and put it on the microscope. But what we did, we will look at the number of sequences in Cyanobacterium in raw water and compared to what was accumulated in a sludge tank after clarification. This is not a sludge from a wastewater treatment plant. This is a sludge from clarification in drinking water plant. And surprisingly, we found lots of cyanobacteria in that in that water. These are only gene sequences, so they don't necessarily mean that the cell numbers were very high. One study we did, and that was very study. This is a work out of SAE Water and a linkage project that was supported by Water RA and obviously ARC, that samples were taken and they were kept in dark. So they were sludge, put them in dark. And we looked at the growth of cyanobacteria. We measured the production of microcystins and also the release of organic matter. If I wouldn't have told you that this is from a sludge, one would look at this particular toxin production and would say, oh, this looks like a culture. You have here the time, days, 16 days, and you have the microcystin concentration. It looks as if something was growing, releasing microcystin a lot. This was not in a culture room under the beautiful light and in presence of, it was in dark, cold sludge. And we look at the same thing for Cyalandus spermopsin production. So we actually think these cyanobacteria grow within the sludge. And that has been the topic of a recent study, ongoing study, that is going on between Vihai, Lower Murray water, and Melbourne water, that we're looking at some sludge and the fate of the cells in the sludge. So, to finish off, I know I've been bombarding you with lots of information about these different projects, but there is a resource that I invite you please to look at them. This is the Water Research Australia Algal Innovation Community of Interest. And if when you go to the web page, I put the link up here. I will put it after us when I stop talking in a minute. I will put the link in the chat as well. And this will give us our current projects and the projects we are moving forward. It's not all our projects, but this covers most of them. And then the ones that are not here, they're at least listed there. And as you can see, these have been done in 4 stages. The goal of this staging is the 3-year program. And we are hoping that we start by detecting them, treating them, actually identify their costs. What we didn't discuss here was the cost of these blooms, and then treat them, and then finish off by training the operate, not only the operators, but the practitioners of the future. Have the future PhDs who will graduate from our amazing universities and will start working on utilities to use these new technologies. And some of them are not new, but they're modified to detect them and remove these from the water. So I again like to thank Brad and his team for inviting me. Thank you all for listening to me.